Good morning, good afternoon, and good evening to all the participants around the world. My name is Huiwen. I'm a PhD student from the University of Queensland Australian Institution for Bioengineering and Nanotechnology. It's my pleasure today to present my PhD project titled Revealing Cell-to-Cell Variability Changes in the Aging Immune Cells by Applying Accurate Gene Expression Variability Metric. To begin with, I'd like to introduce what is the cell-to-cell transcriptional variability and its application in aging. Early in the developmental process, cells are theoretically genetic identical, but they display non-genetic heterogeneity within the cell differentiation process. Just like the ball in the machine on the left, they are all white at the very beginning and gradually change to more and more colorful as they aged. And during our aging, we accumulate molecular damage, and those cells adapt to these changes, which is generally a stochastic process. And cell-to-cell transcriptional variability, also referred to transcriptional noise and many other names, reflects the heterogeneity occurs within the cell population, both intrinsic and extrinsic. Recently, Martinez et al. Performed single-cell sequencing of CD4 plus T cell in old and young mice, where they showed that as mice aged, the expression of genes involved in the pathway corresponding to immune cell stimulation was not as robust and exhibited increased cell-to-cell variability. Although many studies have concluded similar increased cell-to-cell variability changes in a variety of aging tissue and cells, this phenomenon is not universal. Willie et al. Reported a decreased variability genes in the comparisons of senescence and quiescent cells, which is a very powerful and widely used model in aging research. And that is because many metrics have been used to measuring these cell-to-cell variability changes in both bulk and single-cell RNA sequencing data, but the accuracy and consistency remain unclear, and this might lead to a misinterpreting of the further downstream changing directionality. We decide to perform a metrics performance evaluation before applying these metrics to actually quantifying the transcriptional variability within each of the cell types. We applied these comparisons on 12 metrics in 4 main categories and used simulation datasets, control data, and cell type specific tabular mirror SNES datasets from this big database. And also, we're mainly focusing on 2 criteria. One is the consistency of these metric performance, and the second is whether these metrics measurements will be influenced by different data structures. Talking more about the characteristics for the metrics used in the comparison, the first big category of these metrics is the basic statistical dispersion metrics, which including the standard deviation, interquartile range, median absolute deviation, final factor, and the coefficient of variation. The second big category is the local normalized metrics, and this LCV, local coefficient of variation, was first introduced to apply on bulk RNA sequencing data. In detail, this method will arrange the genes based on their mean expression from small to large and apply the starting window default parameter as 500 and reorder the genes within the starting window by HCV value. Then it will give a LCV value for each of the genes based on the quantile of these genes CV within this entire sliding window. In the end, this LCV will be ranging from 0 to 100, which is slightly different from all these other methods. And the other one is the distance metrics for single-cell proteomic data, which they use a CV divided by the median CV of all the genes. The third one is the regression model-based metric. On the right-hand side, you can see that this is basically a generalized linear regression model between the mean and different kind of variation. And the plot here shows the mean versus the variance within the dataset. And this is being widely used in single-cell RNA sequencing normalization methods and many other applications as well. And we use the inverse Gaussian distribution with canonical link functions with second-order polynomial regression to best fit the model. We use the difference between the observed value and versus expected value to represent the variations for each of the genes. And the last one is about the biological variation metric. Here, BASIC is the only one that designed for single-cell RNA sequencing data by using Bayesian statistics. In general, it can help us to identify the biological variation component and the technical variation component. And for downstream analysis, we'll be only focusing on the biological component. Talking about the dataset using the comparison, the first is the simulation dataset. We use a combination of varied biological coefficient of variation component, known as BCV, which represents the within group variability. And there are 4 groups were designed for these comparison, which are high and low BCV, high and moderate BCV, moderate and low BCV, as well as high low BCV with a certain amount of dropout rate. And for the control data, we use the spiking data that was designed by Green et al. 2014 to investigate the noise with different models. And lastly, for the cell type specific data, we applied these large aging data sets, tabular where we use 2 main subtypes from the marrow tissue, which is the mid-naive B cells, and the immortal B cell represents a big sample size and small sample size. And both has been sequenced by FACS water date technology and microfluidic droplet technology. And we use both of them to compare the influence by different sequencing technology. The first mean evaluation criteria is the measurement is estimated independently on the structure of the data, which including the numbers of zeros per gene, the mean expression, as well as the gene length. And the second is to test whether the measurement is agreed with non-biological variation or not. Here we applied the stably expressed genes that was specifically for mouse single-cell RNA-seq data. And lastly is to test whether the measurement is influenced by any of the data types, which including the different numbers of cells in each of the data sets, sequencing technology, and many other aspects. Here is just a glance of the results for the naive B cells, but I have actually applied on all the data sets. And this is just showing you how a poor matrix would perform versus how a good performed matrix would look like. So this is against the numbers of zeros in the data, and we know that it's a big issue for the single-cell RNA-seq data that it has excessive zeros and can influence on many other things. And here on CV, as we can see here, that with more and more numbers of zeros per gene, the CV values tend to become larger, while the LCV, you basically can't see any correlations between the numbers of zeros per gene versus the metric measurements. And we rank 1 for the poor performance and 10 for the best performance. Similarly, we did this for the mean expressions as well. And it's not hard to imagine that similarly for CV, well, we have extremely small value. If you remember the formula, we will get extreme large CV. And this is not represent any of the variation changes, but actually the sensitivity to the low expressed genes. While on the LCV, it's the same as previous. We don't see any correlations at all. Similarly, we also tested for the gene length. And as we know that gene expression is correlated with coding sequencing length, but surprisingly, we don't see any correlations or strong correlations between any of these metrics. In fact, we could conclude that these gene length is not affecting or largely affecting estimating the transcriptional variabilities level in each of the cell type. About the stable expressed genes that I mentioned previously, and this gene list was extracted from Lin et al. 2019 paper, where they demonstrate that SCGs identified from single cells based on their stability indices are considerably more stable than housekeeping genes defined previously from cell populations across diverse biological systems. And this is shown in Heatmap MAD and also the EV log CV square metric, where we actually plot the distribution for each of the measurements, and the red dot represents these SCGs, and ideally like these SCGs fall in the 4th quantile range, as what these would do in the EV log CV square. And this is what we expected that a good matrix would perform. Once we have these 4 different aspects, we then checking the consistencies for different metrics performed on different scenario. And these consistent one represents the difference between a large sample size, which is a naive B cell around 300 cells, while a small sample size, which is a immature B cell around only 40 cells. And we would like to see the difference between the correlation for each of the metrics and whether the metrics performance is consistent and can handle with different numbers of cells in different data sets. Same result, same analysis was performed to test on the consistencies on different sequencing technologies and different levels of variation, where we use a normal single cell RNA-seq data versus a spiking data, and also the variations that occurred in the benchmarking data set, which is the simulation data set that I have been introduced previously. In the end, we come up with such a heat map to show how metrics of gene expression variability performed across multiple criteria. One thing I want to highlight here is for LCV. If we're looking at the ranks across all the criteria, it actually performed really well, but it shows an A in the SEGs as it's internally filtered out where extreme values from both ends at the extremely stable or extremely varied, and this might end up with a less data set and losing of our target genes. For that purpose, I don't want to lose any genes or any highly or low expressed gene or variable genes. I would use this EV log CV square instead, where it actually gives a very good performance and it can handle with different scenario as well. Next, with these metrics in mind, we like to revealing this expression variability changes in the mouse HSC's lineages. And this is also the data from Tabby Ramirez senescence and in the marrow tissue. And this bar plot actually shows you the numbers of cells in the marrow for different tissue types. And as I mentioned previously, that actually sample sizes are actually affecting on measuring these transcriptional variability accurately. Because of that, we would like to include only cell types that have at least 300 of cells per cell per age group, and that will end up with the HSC cells, granulocyte, and also the naive B cell. And the lineages for each of the 3 cell types has been shown up here in this graph. Then this is the statistical pipeline for testing differentially varied genes and identify those transcriptional variability changes in aging. The first big chunk is to minimizing the effect from technical zeros, and we applied different filters to removing the outliers from the data and only taking those genes that actually have certain expressions for the downstream analysis. We performed a clean test to check for the DV genes per cell type and also consider the expressions based on their mean value. And these Venn diagram down here actually shows you the significant differentially varied genes at different cell types, and also whether they are increased variability during aging or decreased variability during aging. And we can observe that the overlap is really small compared to these aging cell type specific. And this can be concluded in our downstream analysis as well, that these differential varied genes are more cell type specific, and it's showing very cell type specific functionalities as well. And for the downstream analysis, we're mainly focusing on the transcriptional factor networks and also the pathway analysis. So this is showing you the plot for the transcriptional factors co-expression networks in young and old group at naive B cell, granulocyte, and HSCs. And we can observe in these networks that these red dots actually represent T transcriptional factors, and the green ones are the genes. All the circles here represents the significant differentially varied genes during aging, and the circle sizes represent the level of cell-to-cell transcriptional variability levels in each of the age groups. And TFs in the table was ordered based on average mean expression in each of the cell types. One thing I want to highlight here and really interesting to see is that as you can observe, the frequency of each of the transcriptional factors is aligned with their transcriptional variability changes. For instance, for these ETS1 transcriptional factors, it actually has more connections in young group versus old group. And similarly, on the gene expression levels, we would also observe that is a increased variability for these genes at in young group versus old group. And this is showing you the consistent connections changes in aging immune cells. And when we applied the pathway overrepresentation analysis based on the increased variability genes and decreased variability genes, we show that these pathways are very cell type specific, and this is especially shown in those decreased variability genes in aging. And one thing I want to highlight here is about the cellular senescence pathway, which is a very essential pathway in aging research. It has been shown that most of the genes in these pathways has been showing a increased variability characteristics in aging. And it's surprising to see that in our granulocyte and naive B cell is actually follow these similar trend, but HSCs actually performed differently compared to all these others. And if we're digging into all these genes and we plot a PPI network based on the genes we found previously in each of the pathways, we would observe a very similar pattern for the network node degree distribution changes. And you can see that these granulocytes actually have more connection for each of the nodes, and where we observed an increased variability in aging, while HSCs shows a very low kind of changing on the degree of distributions. And this is highly matched to what we observed on the expression levels as well. In conclusions, the first is the metrics is measuring for cell-to-cell variability is depending on the research aims, whether you want to obtain a higher accuracy, which you can apply for these LCV method, or you want more genes included for your downstream analysis. So that's why I chose the EV log CV square metrics for these comparisons. And also our work demonstrates the potential of TFs and the mechanisms that contribute to aging immune cells with the best metric that at measuring gene expression variability. But also identify the significant level of variation changes are not only presenting at gene level, but also at cell type levels. In the end, I'd like to thank everyone from our lab for their consistent help and support, and also from our collaborators, Professor Youngvik and Dr. Sheldon, for their insightful ideas. Thank you everyone for listening, and I will take questions.